All right, so myostatin is a molecular brake that prevents excess growth of muscle. And so the question is really, how does myostatin work, and what is it as a molecular structure? So what myostatin is a member of this huge superfamily of growth signaling proteins that are circulating proteins that are part of the transforming growth factor beta or transforming growth factor family. And so what it is it's a family of over 30 proteins made up of TGF-beta 1, 2, and 3, the bone morphogenetic proteins that are again important in bone formation as well as other components of that, the activins, the inhibins, so these are two different proteins that are important in development. The growth and differentiation factor, again, as it says, these are important in cell growth and then the differentiation of cells down different lines. And then the glial-derived neurotrophic factors, again, which are going to be things that are important in maintaining or growing nerves within the brain and other structures. And then this Mullerian inhibitory substance. So myostatin is a member of this family. It is also known as growth and differentiation factor 8. So all it is really is a growth and differentiation factor. So it's one of this large family of proteins that can circulate throughout the body, and it can bind to receptors and signal into cells to induce either growth, differentiation, or transformation. So from one cell type into another, or cause it to go from a non-differentiated cell, so like a stem cell, into something that's more of a tissue-specific, or it's a differentiated cell. All right, so myostatin itself, like all of these TGF-beta family members, works by binding to a receptor tyrosine kinase, and specifically myostatin binds to activin, and it really binds to an activin receptor called activin receptor 2B, and it also binds to an ALK. So it's an activin receptor-like kinase. So this is where, again, as with IGF-1, where it brings two different receptors together and one phosphorylates the other, it's essentially what happens here. So when myostatin binds to the activin receptor 2B and the ALK receptor, what happens is it brings the two of them together. The activin receptor can activate this ALK receptor, and now the ALK is an active kinase. And so when ALK is activated, what it does is it's going to signal through these proteins called the SMAD proteins. And SMAD is an acronym where you smush together the genes that they were discovered in C. elegans, or these little worms, and those were the SMA genes. That's what they were called in the worms, and the ones that were discovered in Drosophila, which were called the MAD genes, the mothers against pentapedic. And so basically these were two different developmental gene families that were discovered, one in worms, one in flies. When they realized, oh, you know what, these are the same proteins, they're the same family, they just took them together and they smashed them together into the SMA. The MA was in both of them, so they just put them together into the SMADs. All right, so now what we've got is we've got these growth factors that bind to a receptor. They bind to the receptor. Once the receptor binds to the growth and differentiation factor, it can now signal that there's a growth and differentiation factor protein there, and it's going to signal to these SMAD proteins, and the SMAD proteins can then move into the nucleus where they're going to change transcription. And that's, again, primarily what myostatin is able to do. So the structure of the signaling pathway is actually quite short given a lot of the things that we've looked at. So myostatin can circulate in the body, is produced by muscle, and it circulates throughout the body. And what it is, it's thought to be a protein, this kind of protein that basically tells you how much of that cell type you have in your body. And so the idea is that you've got a pre-programmed level of muscle in your body. And each muscle cell is producing 1 unit of myostatin. And when you get up to that threshold of having enough of muscle for your genetics, now you've got so much myostatin, because each cell is producing its 1 unit, that's now going to feed back by binding to the receptor, and it's the activin 2B receptor and this ALK receptor. And then there's 3 different flavors of the ALK receptor that it can bind to, but all you have to know is that it binds to the activin 2B and the ALK receptor. And when the ALK receptor and the activin 2B are together, the ALK receptor becomes active, and it phosphorylates SMAD2 3 in this case. And when SMAD2/3 is phosphorylated, it changes its shape. And that change in shape allows it to interact with or bind to this co-SMAD called SMAD4. And what SMAD4 is, it's the chaperone that basically is going to take the SMAD2/3 into the nucleus and allow it to be a transcription factor. So the way that this process works is that myostatin binds to the receptor, and the receptor can then phosphorylate SMAD2/3, change the shape of SMAD2/3, and when SMAD2/3 is in this novel shape, SMAD4 binds to it, and it can now move into the nucleus, and it can bind to and change transcription. There's a couple of ways that you regulate this, or there's a number of ways that you regulate this pathway. The first one is this protein called follistatin. And follistatin is a broad TGF-beta inhibitor. So what it can do is it can bind to all the different forms of TGF-beta within the body. So if you have TGF-beta 1 or TGF-beta 2 or growth and differentiation factor 11 or myostatin, growth and differentiation factor 8, follistatin can bind to it, and when follistatin binds to it, it can't bind to the receptor. So that's one way that you can block myostatin activity. The other thing is that myostatin, like most exported proteins, are produced in this long pre-pro protein form. And then as it gets exported, you cut off the pre and pro peptides. And when you do that, those things can circulate as well. And so you can actually use the myostatin propeptide as a way to block it, because when it's still got the propeptide with it, it can't bind to the receptor. So if you wanted to inhibit myostatin, you can inject the propeptide of myostatin. It could cover up the myostatin and you wouldn't be able to signal here. And then as we go through this, the other thing you see is that there are these proteins down below. They're called ski, snow, and notch. And ski, snow, and notch are transcriptional repressors. And what they do is they can repress or prevent SMAD2 or 3 from doing its job transcriptionally. Okay, and what SMAD2/3 can do is it turns off growth genes, or it turns on genes that inhibit growth. So it can turn on genes that are important for things like muscle atrophy. All right, so there's a number of different ways that you can regulate this pathway. And again, that suggests that this pathway is really important for regulating something significant. And we saw that by just looking at the pictures of the animals, that the significant effect is that you take a control mouse, that's this cute little sucker here, and when you put in a dominant negative activin 2B receptor, so it can't bind to myostatin, you get a bigger muscle mass. When you put in the follistatin, so instead of putting in something that can't signal within the muscle, now you put in follistatin, now you get a huge mouse. Big old swole mouse, it just goes around the cage, and you'd think that this would be the dominant one. Interesting thing happens. This is just a side fact, that if I take this little tiny mouse and I take this big huge muscular mouse and I put them both into the cage, what's going to happen is this big huge muscular mouse is going to go into the corner of the cage and start shaking because it's afraid of the little tiny mouse. And it's got what we call big man's disease. Big man's disease is, we had always thought that you're really big when you're young. Your parents always say, play gentle with everybody. You don't want to hurt anybody. So you're really kind of timid and you're scared and you don't want to do anything because you've been always told since you were a kid that you're bigger than everybody else. You have to play nice. But apparently you don't even need that feedback. Apparently just by being a big, huge, strapping individual, somehow this affects the brain as well as affecting muscle mass. So these animals tend to be very timid and quite shy. All right, but so what you can see is that if you put in any of these things, so if you break the receptor for myostatin, you get bigger muscle mass. If you put in follistatin, you get bigger muscle mass. And it's not just that you get bigger muscle mass, it's that here's the propeptide to myostatin, you get probably about a 2-fold increase in cross-sectional area of the fibers. Dominant negative activin 2B, you get a nice increase. Again, with follistatin as well. The other thing that you get, interestingly, with myostatin in these models, because the knockout occurs, or the overexpression of follistatin or the propeptide occurs before the animal is actually born, you actually get more muscle fibers. So what it's telling us is that myostatin controls two things. It controls the size of the fiber once you have the fiber, but it also tells you how many muscle fibers to make in the first place. So developmentally, when you're in utero and you're still in your mom, basically this growth factor starts to circulate and it says, we've made enough muscle cells, let's stop making more muscle cells. So if you don't have myostatin signaling in utero, and there's at least 2 boys who have been born that we know of that have been born without myostatin, what happens is they get more muscle cells to begin with. So yeah, their muscle fibers are bigger, but they also have more muscle cells. So the result is a significantly bigger muscle mass. Just like myostatin, follistatin, the propeptide, if you put in this protein, this transcription factor SCI, now what you do is you take your muscle fibers from this and you make them into this. So they're huge. They're about a 3+ fold increase in muscle fiber size. So again, if you put in SCI or SNO, or you put in Notch, or you put in follistatin, or you break the activin receptor, or you put in the myostatin propeptide, now what you've done is you've blocked the myostatin pathway and you get this nice huge increase in muscle fiber size. And again, it doesn't matter where along the pathway you block it, blocking it at any point is going to result in this huge increase in muscle fiber size. So the way that this works is essentially that myostatin controls fiber number, but that only happens in development. So if you are lacking myostatin in utero, your muscle cells continue to proliferate much longer than an average or a non-mutated or a non-knockout individual, and you get many, many more muscle cells, and so you've got more muscle cells when you're born. And then after you're born, myostatin controls muscle fiber size. And what it does to do this is it regulates genes that are important for growth, and what it does is it blocks the genes that are important for growth, or it stimulates the production of genes that are important for breaking down muscle so that you can regulate the muscle fiber size. But the primary thing that it's doing is it is increasing muscle cell number. The second thing that it can do to control muscle fiber size is by regulating mTOR activity. And we'll see that you can do this in adult animals so that even when you can't get an increase in muscle fiber number, you can still get an increase in muscle fiber size simply by activating transcription and regulating mTOR activity.